Okay, let's dive in. There's this fundamental mystery in biology, almost like a black hole in life's history.
You mean how we got from simple cells, prokaryotes, to the complex ones, eukaryotes?
Exactly. How did that sudden jump happen with all the internal bits, the nucleus, mitochondria? It's a huge leap scientists have puzzled over for, well, ages.
Yeah. And we have the endosymbiotic theory, which is, pretty well accepted now. The idea of cells merging.
Right, one ancient microbe engulfing another.
But the steps leading up to that big event,
Yeah.
that's always been murky. Like, we see the start and the end result, but the middle is kind of fuzzy.
The missing frames, exactly. But look, there's some really exciting new research just published in PNAS.
Ah, the paper from that international team.
Right.
Baines, Valencia, Madrid, Zurich.
That's the one. And it offers a completely different angle on this whole transition. So our mission today, listener, is to really dig into this study. We want to understand how the basic instructions of life, the genetics, might have changed to allow for this, well, explosion of complexity.
Focusing just on this PNAS paper, then. Getting to the heart of their argument.
Precisely. Let's unpack their key insights.
Okay, so where do they start? It seems to begin with something fundamental.
Yeah.
The length of genes and proteins.
Yeah. They analyzed a truly enormous amount of data. What was it? Nearly 10,000 proteomes.
And over 33,000 genomes.
I think-
That's just staggering. Across all domains of life.
And they found this really consistent pattern. Both protein lengths and the gene lengths follow these log normal distributions.
That's right, log normal. And this holds true basically everywhere, from bacteria up to complex animals.
Okay, hold on. Log normal distributions. For those of us not immersed in statistics daily, what does that actually tell us? Why is that significant here?
Good question. Well, typically when you see a log normal distribution, it points towards processes driven by lots of small multiplicative factors acting together.
Multiplicative, like things compounding, not just adding up.
Exactly. Think of growth rates, maybe, or processes where each step depends proportionally on the current state. The researchers saw this universal pattern and thought, okay, using Occam's razor.
The simplest explanation.
Right. The simplest model is that gene length evolution is driven by some kind of underlying multiplicative stochastic process. Random genetic events, insertions, deletions, duplications, whatever happening over time, their effects multiplying.
Okay, so a fundamental pattern suggesting a common underlying mechanism for how gene lengths change across all life. Interesting. What did they look at next?
They then tracked how the average gene length changed over evolutionary time, starting way back, conceptually at least, near LUCA.
The last universal common ancestor, our earliest ancestor.
Exactly. And what they found, both from their theoretical models and looking at the actual genomic data, is that average gene length seems to have increased exponentially since then.
Exponentially. So a steady, accelerating increase over billions of years.
Pretty much. And they also found this interesting scaling thing, a scaling invariant mechanism. Basically, the amount of variation, the spread of gene lengths around the average, scales proportionally with the average itself.
Okay, so the pattern of variation stays consistent relative to the average size.
Yes. And perhaps less surprisingly, but still important, they confirmed that average gene length is actually a pretty good proxy for an organism's overall complexity.
Which makes intuitive sense, I guess. More complex organisms may be needing longer genes on average.
It holds up across thousands of species they looked at. It's a strong correlation.
Okay. Now, let's talk about prokaryotes versus eukaryotes. In bacteria and archaea, the simpler cells, there's a tight link between how long a gene is and how long the protein it makes is.
Right. Very tight. Because prokaryotic genes are mostly coding sequence. There's very little non-coding DNA, introns and such, compared to eukaryotes.
Well, if the gene gets longer, the protein almost automatically gets longer too. They sort of evolve hand in hand.
In lockstep, yeah.
But the research points to this really dramatic moment, a kind of breaking point. They identify a specific threshold.
Right. When the average gene length hits about 1,500 nucleotides, that seems to be a critical number.
And what happens then?
That tight coupling breaks down. Suddenly, average protein length seems to hit a ceiling, stabilizing around 500 amino acids or so.
Even though the genes keep getting longer.
Exactly. The average gene length continues its exponential climb in eukaryotes, but it's now decoupled from average protein length. And this happens right around the time eukaryotes appear in the fossil record and in molecular clock estimates.
So that stabilization of protein length while gene length keeps growing due to non-coding bits, that's the signature of eukaryogenesis in their model.
That's the core observation. The decoupling marks the transition.
Which leads them to this really cool, maybe slightly mind-bending concept. And An algorithmic phase transition. That sounds very physicsy.
It absolutely is. They explicitly draw that analogy. Think of water freezing into ice, or a magnetic material suddenly becoming magnetized below a certain temperature, a phase transition.
A sudden qualitative change in the system state when a parameter hits a critical value.
Precisely. And they argue that life underwent a similar transition when average gene length reached that critical point, around 1,500 nucleotides. This event, eukaryogenesis, fundamentally divides life's history.
Into what? Two phases?
Yeah, what they call a coding phase, dominated by prokaryotes where gene length and protein length are linked, and a non-coding phase, characteristic of eukaryotes, where they're decoupled thanks to introns and other non-coding sequences.
And they even see other signs, like in physics. You mentioned something, critical slowing down.
Yes, that's another hallmark of phase transitions in physical systems. Right near the critical point, the system can become sluggish, taking longer to settle down after perturbations. It sort of hesitates.
And they see evidence of that biologically.
They found signs of this critical slowing down in the genomes of early diverging eukaryotes, like some protists and fungi, suggesting these lineages might have been closer to that transition point, still sort of working out the kinks of the new system.
Fascinating. Okay, so we have this transition triggered by gene length, but why call it algorithmic? What's the algorithm that's changing here?
Right, this is the really clever part of their argument. Think about the coding phase, the prokaryotic way. Making a protein is basically read the gene, translate it. Longer gene, longer protein.
A direct algorithm.
Pretty much. But as genes kept getting longer and longer, trying to find new, functional, even longer proteins within that direct system became, well, computationally very difficult.
Like searching for a needle in an increasingly massive haystack. Finding functional sequences gets harder.
Exactly. The search space explodes. It becomes algorithmically complex, maybe even intractable, to keep efficiently exploring longer protein possibilities just by making genes longer and longer in that direct coding way. The system was hitting a complexity bottleneck.
So evolution was sort of painted into a corner by its own success in lengthening genes.
You could put it that way. There was tension. And their proposal is that the invention of introns and extensive non-coding sequences was the solution. An abrupt one.
How did that solve the algorithmic problem?
It allowed for the evolution of the spliceosome, the machinery that cuts out introns, and the nucleus, which separates transcription and splicing from the final step, translation.
Okay, separating the steps in space and time.
Yes, and that separation, plus the modularity introduced by splicing different exons together, fundamentally changed the algorithm for finding new proteins. It broke the linear relationship, it made the search process nonlinear, much more flexible, and arguably more computationally efficient for exploring complex protein structures.
So introducing introns and splicing wasn't just adding complexity. It was a way to manage complexity, a new, more sophisticated algorithm for generating protein diversity.
That's the core idea. It reduced the computational burden and opened up vast new possibilities for protein evolution, enabling greater complexity.
Wow. And they put a date on this algorithmic revolution.
Based on their analysis, calibrating with molecular clocks and the fossil record, they estimate this phase transition. The dawn of eukaryotes happened roughly 2.6 billion years ago.
It really reframes the origin of eukaryotes, not just as acquiring new organelles, but as a fundamental shift in information processing at the genetic level.
Absolutely. It connects the molecular level, gene lengths, coding versus non-coding DNA, directly to this massive leap in organismal complexity.
And the implications seem pretty broad, don't they? This feels like it touches on evolution, computation, maybe even physics concepts.
Definitely. It's a beautiful example of interdisciplinary thinking, bringing ideas from computational complexity and physics into evolutionary biology. They even suggest it might spur new thinking about, say, energy constraints or information theory in evolution.
And you have to think, this event, this transition, it wasn't just about making a new type of cell. It was the enabling step for almost everything that followed, complexity-wise.
Totally. Once you had the eukaryotic cell, with its potential for compartmentalization, regulation, and complex gene expression, that paves the way for multicellularity, for sexual reproduction, for complex development, eventually even for things like consciousness and societies.
It really underscores how pivotal that moment was. Without that algorithmic shift 2.6 billion years ago, the whole trajectory of life would likely have been profoundly different.
It's hard to even imagine, really.
Okay, so let's try to crystallize the main takeaway from this deep dive. The PNAS study suggests the emergence of eukaryotes wasn't just slow and steady tinkering.
No, it proposes it was more like a fundamental shift, a phase transition,
Driven by the relentless increase in average gene length over billions of years.
which eventually hit a kind of computational wall, making it hard to keep evolving longer, more complex proteins efficiently within the old prokaryotic framework.
Until a critical threshold around 1,500 nucleotides in average gene length was reached.
Triggering this transition, marked by the decoupling of protein and gene length evolution, thanks to the widespread adoption of non-coding DNA, introns, and the evolution of splicing.
Essentially, life adopted a new, more sophisticated algorithm for processing genetic information and generating protein diversity.
An algorithmic phase transition that dramatically reduced the complexity constraints and unlocked the door to the eukaryotic world.
Right. And thinking about that phrase, algorithmic phase transition, it really sparks a thought, doesn't it? Could we look at other major leaps in evolution or maybe even big shifts in human technology or society through a similar lens?
That's a fascinating question to ponder. Are there other instances where increasing complexity in one dimension forces a system to find a fundamentally new way of organizing or processing information to overcome a bottleneck?
Could similar principles be at play? Thresholds of complexity leading to abrupt qualitative changes in how systems operate, whether biological or technological.
It certainly makes you wonder about the underlying rules governing complexity and innovation across different domains. Are there universal patterns?
Definitely food for thought. It just highlights, again, how incredibly profound that single evolutionary event, the rise of the eukaryotic cell, truly was for everything that came after on this planet. Something to keep turning over in your mind.